Okay, welcome everybody. This is the lab test review for the urinary system. This material will be covered on Saturday's lab practical, so be sure and listen up. Again, the standard precautionary message indicating that you do not want to use this as a substitute for studying the models that are available in Biosis or in the lab during the lab period time designated. And with that said, let's begin with our first model. Okay, our first model is asking you to identify the cortex and the medulla on this model of the kidney. Okay, so the cortex is going to be the layer that lies just underneath the capsule and superficial to the medulla. So here, it's this dark area that I'm tracing right now with the pointer. Okay, this is cortex. Now, where is the medulla? The medulla is this underlying layer where the renal pyramids are. So the area that I'm tracing around here, this would be medulla. Okay. Next question, identify the regions indicated on this model. Okay, so let's start with A. A is this small cup-like structure into which the renal pyramids drain. Actually, let me correct that. A appears to be pointing to this little open area right here. Okay, this little open area with the holes in it, these are the renal papillae, and this is where the urine comes out and drains into the structure indicated by C here, which is the minor calyx. Okay, minor calyx is Latin for little cup. B here is pointing to a renal column. The columns are basically tiny pieces of cortical tissue that lie between the pyramids. So technically you could call the renal columns part of the cortex, although in practicality they don't contain significant amounts of filtering units of nephrons. They just contain primarily veins and arteries that move through the kidney and eventually take the bloods to the filtering units, which are called the nephrons. So C is a minor calyx, A is a papilla, B is a renal column. Okay, now what is D? D is the renal sinus. The renal sinus is a big open area inside the renal pelvis. The renal pelvis would be this entire anatomical structure that I'm tracing, and this is where the urine drains from the major calyx, major calyces, into this open area before it goes into this area down here, which would be the ureters. Okay, more structures to ID. What is A pointing to again? Again, A is pointing to a papilla, okay, a papilla. This is at the tip of the pyramid, the apex of the pyramid. You can't see this particular tip of the pyramid because the pyramid for this would be going into the slide, deep into the slide. Okay, C here is pointing to, again, a minor calyx. B is pointing to a renal pyramid. So in this particular model, we can see 1, 2, 3, 4, 5 renal pyramids. D, again, is pointing to the renal pelvis. Okay. E is the ureter, and that takes care of all the structures on here. Over here, what are these pointing to? These are renal papillae as well. Okay, notice the tips of the pyramids. If you look very closely in a real kidney, what you'll see is that they have a lot of tiny holes in them, and that's where the urine drains from the pyramids through these tiny holes into the minor calyces, from there into the major calyces, and from the major calyces into the renal sinus, and from there into the ureters. Okay, another model. Let's start with A. Okay, what area is A pointing to here? Okay, I'm tracing it here and I'm tracing it here, and this is cortex. Okay, this is cortex. This little dark area here, this would be capsule. Okay, that I'm tracing right now. What is B pointing to? Okay, B is pointing to this area here. Okay, in the low magnification, and this area here in the high magnification, and that is the medulla. Okay, the medulla is populated again by renal pyramids. C, pointing to this collection of structures here, 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 and here. This is the nephron. This is the unit of the kidney that actually cleans the blood. Okay, moving down. D, again, is pointing to the renal pelvis. E is pointing to the ureter. F, down here, is pointing to a major calyx, And then, further down, G is pointing to a minor calyx. So let's find the minor calyces here. There's one, there's one, there's one, there's one, there's one. Okay, those are all minor calyces. H is pointing to the renal papilla, okay, the tip of the pyramid through which the urine drains. So let's find all the renal papillae on this model. There's one, 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 and there's one, okay? All right, moving on. This is the dark torso model. We've removed a lot of the internal organs in the abdominal pelvic cavity so that we can see most of the urinary system. What is A pointing to here, this dark area on the outside of the kidney? This is the capsule. This is the capsule, okay? What is B pointing to? B is pointing to a ureter, okay? C is pointing to the bladder. So how is urine made? Urine is made as a result of the filtering of blood that comes into the kidney through the renal artery. The filtrate is adjusted for waste content and water content. The nutrients that are in the blood enter the filtrate but are eventually recovered and sent back into the blood, so that by the time the filtrate is ready to enter the minor calyces, it contains primarily nitrogenous waste products, water, electrolytes, and in some cases creatinine, and in some cases uric acid. Okay, so urea, uric acid, creatinine, water, and electrolytes, things that you're supposed to find in urine. Things that you're not supposed to find in urine include things like blood and sugar and anything living, right? No bacteria, no parasites, no protozoans, no fungi. If you have those in the urine, it'll appear cloudy, and that's an indication of a UTI, or a urinary tract infection. So as the filtrate is made inside the kidney, there's one kidney, there's another, okay? This is the diaphragm up here, okay? What happens is that it enters the renal pelvis, drains from there into the ureter, and then from the ureter it drains into the urinary bladder, and once the urinary bladder fills up with enough urine, the urinary bladder will contract. You will at the same time relax your external sphincter. Your internal sphincter will relax for you. The internal sphincter is made up of smooth muscle, and it encircles a structure called the urethra, which is the tube that connects the bladder to the outside world. And at that point you will urinate. That process is called micturition. Okay, this is an actual kidney, and what they've done here is they've shown you a full kidney, and they've cut a kidney in half for you to see. And what A is pointing to, this is capsule. See this sort of cellophane-like wrap that surrounds the outside of the kidney. That's capsule. B is pointing to cortex. Cortex will have a granular appearance because of the abundance of Bowman's capsules in the cortex. Bowman's capsules are the first part of the nephron. And then C is pointing to a ureter. That's a ureter on a real kidney. And D here is pointing to the medulla. Now notice that the medulla, in the models that we've looked at and also in this actual bisection of the kidney, has a striped appearance as opposed to the cortex, which has a granular appearance. And the reason for that is that the medulla contains structures called the loops of Henle. There's millions of these loops of Henle that descend deep into the medulla, and running parallel with them are the vasa recta. The vasa recta are the blood vessels that exchange components with the filtrate, which is contained inside the kidney tubules. And also you have collecting ducts, which is where the distal convoluted tubule empties its filtrate for its final trip through the medulla, and eventually out the ureter into the bladder, and then through the urethra and out the body. So that parallel arrangement of these structures gives the pyramids a striped or a striated appearance, and that's also what gives the cortex a granular appearance because of the random arrangement of all these Bowman's capsules here in the cortex. So that's an actual kidney. Your kidneys are about the size of a bar of soap, and they filter all your body fluids every day. That's a tremendous job for something that's so tiny. Okay, what you're looking at here is the functional unit of the kidney. This is the nephron. Now the nephron has several parts to it. The initial part of the nephron is called the Bowman's capsule. Now the Bowman's capsule is tissue that surrounds a cluster of capillaries called the glomerulus. So inside the Bowman's capsule is the glomerulus, and then the Bowman's capsule surrounds that, and this whole thing is called a renal corpuscle. Now, the way that blood gets into the glomerulus is through the afferent arteriole, this little blood vessel right here that branches off the interlobular, or in some textbooks they call it the radiate artery. Once the blood is in here, the powerful hydrostatic pressure that comes from the beating of the heart, combined with the osmotic pressure that's present in the filtrate and in the blood, serves to drive fluid out through the walls of these capillaries and into the Bowman's capsule, and at that point we have formed filtrate. Okay, so what B is pointing to here is the glomerulus. Okay, A is pointing to, if we follow, here's A, let's follow where A goes. A goes back here, and A eventually empties into the collecting duct, and that's how we know that A is pointing to the distal convoluted tubule. C over here, okay, this little wrinkly tube, that is the proximal convoluted tubule. Now the reason that they have the names distal and proximal is basically their relationship to the renal corpuscle. If we were to take the nephron and stretch it out in a straight line, you would find that the proximal convoluted tubule is closer to the renal corpuscle than the distal convoluted tubule is. So that's where the name comes from. D here is pointing to the collecting duct. Many distal convoluted tubules empty into the collecting duct, and the collecting duct eventually empties out the papilla of the renal pyramid into the minor calyx, and at that point we have introduced the urine into eventually what will be an evacuation system. E here is the ascending limb of the loop of Henle. Okay, so if we follow here, what you see is that as the filtrate is produced in Bowman's capsule, it flows into the proximal convoluted tubule, so follow the cursor here, and then from the proximal convoluted tubule it goes into the descending limb of the loop of Henle, which goes down into the medulla, like we're doing right now, and then at the bend in the loop we head back up towards the cortex, and this is the ascending limb of the loop of Henle. Okay, and eventually we get back to the distal convoluted tubule, which eventually leads back into the collecting duct. Now, your kidney has millions of these. Most of the nephrons in the kidney are what we call cortical nephrons, and they're primarily tasked with detoxifying the blood. So they remove nitrogen-containing waste products, drugs and their metabolic byproducts, and other toxins in order to clean the blood. The rest of the nephrons, the other 15%, are what are called juxtamedullary nephrons, or JGM nephrons. And the reason that they're called this is because their loops of Henle are very, very long. They go all the way down to the apex of the pyramid, basically to the renal papilla. And their purpose primarily is to regulate fluid balance in the body. And the way they do that is by producing a concentration gradient in the medulla of the kidney. They wouldn't be able to effectively do that if those loops didn't descend all the way to the apex of the pyramid and back up towards the cortex. These cortical nephrons have very short loops, and that's why they only contribute a very small amount to the concentration gradient that's in the medulla of the kidney. Okay, moving over here, we're pointing at some of the blood vessels that surround the nephron. So if we start with A, okay, A is a peritubular capillary. Okay, well, what the heck does peritubular mean? It means it surrounds the kidney tubules. What are the kidney tubules? The PCT, the DCT, the loop of Henle. Those are the kidney tubules. And so their close association with these kidney tubules makes it real easy for them to pick up useful solutes from the filtrate, and this is a process called reabsorption, and also to dump waste products into the filtrate, and this is called secretion. Okay, B is basically another branch of a peritubular capillary. These capillaries wind all around the ascending and descending limbs of the loop of Henle and all around the DCT and the PCT. C, over here, is called the interlobular artery. It branches off of E, which is the arcuate artery. The arcuate artery basically runs around the base of the renal pyramid, and it gives off hundreds of these branches, these interlobular arteries, also called radiate arteries in some textbooks. The radiate arteries produce many, many branches of afferent arterioles, which enter many, many different glomeruli. Okay, so the blood flow into the kidney in this region goes from E to C to the afferent arteriole to the glomerulus, and then out this vessel, which is the efferent arteriole, and then to the peritubular capillaries. Okay, moving on. This is just another look at the same model. A is pointing to the efferent arteriole. Blood leaving the glomerulus flows out through the efferent arteriole. The way to remember this, think E for exit, E for efferent. B is pointing to the afferent arteriole. Blood comes into the glomerulus through the afferent arteriole, enters the glomerulus, and this is where the filtrate is made, and C is actually pointing to the glomerulus. Okay, this is another model. This is a look here at some kidney structures. On the left, what you see is a renal corpuscle. And what A is pointing to here, this is the afferent arteriole. Okay, this is the blood coming in to Bowman's capsule. Okay, here's Bowman's capsule. B is actually pointing to the distal convoluted tubule. Now, it's kind of weird, but if you look at the right panel for a minute, I want you to notice something. Okay, notice here in this nephron, there's the proximal convoluted tubule, there's the descending limb of the loop of Henle, and then here's the ascending limb of the loop of Henle, and here is the distal convoluted tubule. Now notice that the distal convoluted tubule bends up real close to the Bowman's capsule on the back end. And there's a purpose to this. The purpose to this is so that the juxtaglomerular apparatus, which is a special set of cells that line this region of the DCT and this region of the afferent arteriole, can talk to each other in order to regulate glomerular filtration rate and blood pressure. So they talk to each other. Basically, one of the things that's monitored by these cells is the solute concentration in the filtrate that's in the distal convoluted tubule, and we measure how much solutes are dissolved in that fluid per unit volume, and we adjust the glomerular filtration rate accordingly. And one of the ways that we do that is by either widening the afferent arteriole or constricting the efferent arteriole, or doing the opposite, okay, depending on the solute concentration inside the glomerulus. Basically, if we open the afferent arteriole and close the efferent arteriole, we increase the glomerular pressure, we increase the glomerular filtration rate, we make more filtrate, and the result is to dilute the filtrate and reduce the solute concentration. If we do the reverse, if we close down the afferent arteriole and open the efferent, what happens is that we lower the pressure inside the glomerulus, we reduce the glomerular filtration rate, and we cause the solute concentration and the filtrate in the distal convoluted tubule to rise. Okay, so it's basically closing and opening the ingoing and outgoing doors to this cluster of capillaries that regulates the pressure in the capillary, and that in turn regulates the speed with which we make filtrate, the speed with which we make pee. Okay. All right, so A is pointing to the afferent arteriole, B is pointing to the distal convoluted tubule, and C is pointing to the efferent arteriole. So this is how the blood leaves the glomerulus. D is pointing to the glomerulus itself. All these blood vessels taken as a collected unit, this is the glomerulus. Okay, so I'm tracing the glomerulus for you here. Okay. E is the proximal convoluted tubule. Okay, Bowman's leads right into the PCT, and F is Bowman's capsule. All right. G is pointing to the arcuate artery. Okay, we're over here in the other panel now. This is the cortex, remember. This is the medulla. This is the base of the renal pyramid. This is the arcuate artery. H is pointing to the collecting duct. Okay, see how all these nephrons jack into the collecting duct. I is pointing to the descending limb of the loop of Henle. Again, there's Bowman's, there's the PCT, there's the descending limb of the loop of Henle. Okay. J over here is pointing to the proximal convoluted tubule, and that's pretty much almost everything here. K is pointing to the glomerulus, and L is pointing here to the distal convoluted tubule, which leads directly into the collecting duct. So just for fun, let's retrace the route that urine takes as it's made. The filtrate is produced in Bowman's, goes through the proximal convoluted tubule, goes down the descending limb of the loop of Henle, goes up the ascending limb of the loop of Henle, enters the distal convoluted tubule, enters the collecting duct. Now where does it go next? All the way to the apex of the renal pyramid, out the top of the renal papilla, which if you look at it real close looks almost like the top of a salt shaker, and into the minor calyx, from the minor calyx into the major calyx, from the major calyx into the renal pelvis, from the renal pelvis into the ureter, from the ureter into the bladder, from the bladder into the urethra, and from the urethra into the urinal. And that's the whole trip. Okay? All right. Here we have kind of an unusual panel, and it's asking you which of the sites here are major sites of filtration, reabsorption, and secretion. Okay, and so I want to be very careful about this on the practical because there's a simple answer to this question and there's a more complicated answer. So the simple answer for filtration is A, okay, and that goes whether we're trying to be complicated or whether we're trying to be simple in terms of what processes happen in which part of the nephron. A is the glomerulus, A is where the filtration takes place. Reabsorption, okay, well, if we want to be completely accurate about where reabsorption takes place, it can take place in D, it can take place in B, and it can take place in C, okay. The simple answer to the question of reabsorption is that it takes place in over here in the distal convoluted tubule here. All right. If you look for the distal convoluted tubule, there it is. Okay. I'm sorry. Back up. The simple answer for reabsorption takes place in the proximal convoluted tubule here in B. Okay. And that pretty much takes care of most of the reabsorption. Okay, that's where most solutes are put back into the blood. Basically, the good stuff is recollected and put back into the bloodstream. Okay, that's the simple answer, B. Okay, the more accurate answer is that reabsorption happens in both D, B, and C. Well, what are D, B, and C? Well, D is the ascending limb of the loop of Henle. B is the proximal convoluted tubule, and C is the collecting duct. Okay, now how about secretion? Well, the complete and complicated answer is that it happens in E, B, and C. Okay, the simple answer is that the bulk of secretion happens in the distal convoluted tubule. Okay, and so if we follow back here, the distal convoluted tubule is E. All right, so what would I suggest that you do on the practical? What I suggest you do on the practical is go with the more complex answer, and I will take that into account when I grade it. Okay. All right. This is a microscopic slide of kidney tissue. We want to identify which is which here. We want to ask the question, which area is the medulla and which area is the cortex? And the answer is that A here that I'm tracing, this is cortex, and B here, this is medulla. Okay. How do we know that A is cortex? Well, because of this structure right here, that's a glomerulus. Okay, and the glomeruli are only found in the renal cortex. So simple answer to that question. Next question here, what are we looking at? Okay, A is pointing to tubule cells. These can be proximal or distal convoluted tubules as they come out of the medulla into the cortex and surround this structure here. This whole thing is a renal corpuscle. This is the glomerulus inside, okay, and this is the outer surface of Bowman's capsule. Okay, so B, if we want to be technical about it, is pointing to the glomerulus, and C is pointing to the parietal endothelium, which is the inside layer of Bowman's capsule, the inside layer of Bowman's capsule. And this is where the filtrate actually collects. Okay, this is a urine test. This is a dipstick test, and what it measures is a lot of different things at the same time. The way you use the urine dipstick test is very easy. You just take the dipstick with a gloved hand, you dip it into the urine sample, and then you lay that wet stick on a paper towel and you look at the color change on the stick, and that color change will correspond to something on the side of the bottle, and that color change will indicate which component we're measuring. Okay, so if you look at these dipsticks here, the first box after the control box, which is a little white box here that you can barely see, is the measurement for glucose. Okay, look how brown that is. If you look on the bottle, okay, what you see is that this brown area corresponds to a urine that has a 2 gram per deciliter glucose content, and that is an indication of glycosuria. Glycosuria could be an indication of type 1 or type 2 diabetes, among other possible metabolic disorders. Okay, there are other things that the dipstick checks for as well. It checks for the presence or absence of hemoglobin, the presence or absence of sugar, the presence or absence of bilirubin, and several other metabolites. Okay, so you can do a lot of quick tests at one time. Okay, asking you, what are these devices? These are urinometers. You would call it a urinometer if there's urine in the cup here, and you would call it a hydrometer if there's water in the cup. And what does it measure? It measures specific gravity. Well, what the heck is specific gravity? It's the density of the solution. And basically what you do is you put this little bobber in here. The bobber has a particular weight to it, and it has a little bit of air trapped inside. And what will happen is that that bobber will sink or float depending on the density of the solution. So you could imagine, for instance, if we were to fill this cup with maple syrup, that this bobber wouldn't sink very far into the solution, and as a result the specific gravity would be very, very high. Okay, so it would be a very dense solution. Over here you've got urine. You can see that the urine has a higher specific gravity than this sample over here, which is distilled water. Look how the bobber sinks almost all the way to the bottom of the cup here. Okay, so what we're measuring is the optical density of urine. That's an indication of what's dissolved in it. Okay, next question here. Which solution has the higher specific gravity, A or B? And the answer is A. See how high the bobber's floating. Okay, as the solute concentration in urine increases, does the specific gravity increase or decrease? And the answer is that it increases. Makes the solution more dense. As a result, the bobber doesn't sink as much. Next question is, are there units for specific gravity? And the answer is no. It is a unitless number because we divide the density of the fluid that you're measuring by the density of water. As a result, the units cancel. Okay, next question. Is the specific gravity of this urine sample within the normal range? And the normal range for urine specific gravity is 1.01 to 1.025, and so the answer here is no. Okay, absolutely not. Okay, next question. Suppose a first urine sample was collected before and a second urine sample was collected sometime after an increase in ADH secretion in the body. Okay, remember, ADH stands for antidiuretic hormone. Would the hydrometer float higher in the first or in the second urine sample? Okay, so we have to think about this. Let's take the first collection. The first collection is going to have a normal specific gravity, okay, something between 1.01 and 1.02 approximately. The second time we collect, what's going to happen? Well, ADH has been secreted in the meantime. What does ADH do? It causes the body to retain water. If the body's retaining water, what's happening to the urine? Well, the urine is becoming more dense because more solutes are in the urine. The result is that the second measurement with the hydrometer would be a higher specific gravity, okay? A higher specific gravity for the urine. And the result then would be a more dense urine, something that's more dense than the measurement of water, okay? There was a block here for pH regulation in the body. Basically, all I have to say about pH is it's a direct measurement of the concentration of protons in solution. A low pH indicates an acidic solution, a pH of 7 neutral, and a pH of higher than 7 indicates a basic solution. The reason that pH is a big deal in the body is because if pH gets too high or too low, we screw up protein conformation, and as a result, the chemical reactions that they mediate will slow down or stop. And we also screw up membrane potential, basically the separation of electrical charge across a living membrane. And the result then would be a cessation of the cell's living processes, so the cell would die. The first places that would probably be affected would be the nervous system, followed shortly thereafter by the musculoskeletal system. The two major regulators of pH in the body are the kidneys and the respiratory system. Okay, the respiratory system primarily regulates pH by controlling the amount of carbon dioxide dissolved in our tissue fluids, and the kidneys regulate pH in a wide variety of ways. The kidneys have the ability to either secrete protons into the filtrate, into the urine, or to retain them as a result, lowering the body's pH and again keeping pH within nice, tight, homeostatic limits. Okay, that's all I have to say for this lab test review. I will see everybody Saturday morning, and have an excellent week.